Good morning. The breakout session and the lunch. Can I ask Hector to join me? We lost our chairperson, so I will take over just a minute. Can I ask Nick to join us? Nick is working for the UK Health Security Agency, and he's going to talk about WEUs for COVID-19 public health decision making in the UK.
Thanks.
Floor is yours.
Thanks, Peter. Yeah, I realised I'd started my talk when I was up here just earlier. A few of the things I said will come up again. So, yeah, I'm going to focus on the decisions made in the UK, really. And I mean, I was involved in presenting to JCVI. I don't know how many times, probably at least 25 times to our committee over the last 3 years. Let's go on to the next slide. Not sure that's working. Okay, so just the key decisions that had to be made. Jumping straight into that, who to prioritise at the start, whether to increase this in the interval between doses, and whether that decision that was really made on without the real-world evidence could be supported and continued. The age recommendation around the Chadox vaccine and with the VITs. Whether and who to boost in the autumns of 2021, spring 22, autumn 23, now spring 23. You know, who should be those groups? Which vaccine should we use? As the new variants came along, was VE high enough against those variants to mean we didn't have to reinstate or we could continue to reduce the NPIs? Yeah, vaccination in pregnancy, and then which vaccines to actually buy to use as the boosters, the monovalents, bivalents, and so on. And then also around the children and whether to vaccinate which age groups and with what as well. So next slide. Oh, it's gone the wrong way. Right, okay. So just to show how these things sort of fitted together within the UK, we have the JCVI as one of the government advisory groups, and also the MHRA expert working group on benefit risk, who the MHRA put together to also have a look at the focus on the safety data as well. The red bits there are sort of information that I was particularly involved in. So the vaccine effectiveness by the different endpoints, looking at our real world data and reporting that into these groups. That also fed into mathematical modelling to sort of project future waves. The COVID incidence itself and severity by age in particular in different risk groups, that was coming from real world data from some of our GP systems. And then the vaccine adverse events, particularly from my side, doing some of the hypothesis testing studies such as the self-controlled case series method to actually assess whether or not the risks that may have come up in some of the for example, in the pharmacovigilance reporting, was substantiated with sort of more detailed studies. So that was all going into these groups, and ultimately they would advise the government, who then would make the decisions based on that advice. They didn't always follow it, actually, but most of the time they did. So I think it has been seen as a success, and I think I'd agree with that, the vaccine programme. I'll focus on the VE and safety in the next slides in terms of how that fed into the decision-making that I've already covered. And particularly they were key drivers in this benefit-risk assessment, along with the actual risk from the disease itself information coming through. Now, one of the key things that we managed to set up that was really important, and Julia here made sure that this information coming from our national register that was set up for COVID was suitable for, and we got the detailed information to do effectiveness and safety. In particular, for example, making sure we did get the manufacturer data through and getting it through rapidly so we could do studies quickly. So this national register, we never had a national register before, was set up for COVID and also used for flu. And I think we're hoping some other vaccines will come into this. That data could then be used for these. And what was one of the really good things about it is it had denominator data. So because it was used as the calling system for the vaccine. So not only could we find out about who did get vaccinated, but we could use our NHS number to find out, to link to it, and then discover they didn't get vaccinated as well. So that enabled us to do the studies. And to say they were very rapid next day extracts. And with a delay, the data also got back into their general practice records. So there were a lot of studies actually that used this NIMS data. We had our Pillar 2 community testing, and that data came through really quickly. Within days, within 5 days, we think we could do an analysis of effectiveness. And then our Pillar 1 hospital testing, there was a little bit more of a delay because we have to wait for people to be discharged from hospital before we can actually find out that they were hospitalised. And then other studies took a bit longer that were based on cohorts within the GPs or care homes, specific NHS worker cohort study that was set up. So you can see these ones took longer to set up and longer to run the analyses for the data to all come together. The ONS did population sampling that also had some results come through, and household contact studies as well, which again took a bit longer to pull that all together, get that information and analyse it. But the first data results came through mid-January, so we were as quick as a matter of 6 weeks after the vaccine first came out. We were able to show, and we were showing it as odds ratios at this stage from our test negative result, saying that after the first dose and then after the second dose, we were seeing a reduced odds of testing positive, so evidence of effectiveness after a single dose, and then that also being maintained and then improving after the second dose. So that was mid-January, and that really fed into JCVI's decisions, particularly this last data point. Once we were beyond 4 weeks, we were 3 weeks after the dose was given, we were seeing, and that would have covered a number of weeks after that, we were seeing that effectiveness. And then as we went over time, now the axis is vaccine effectiveness. We saw that, and looking at both alpha and delta, so alpha in blue, delta in orange, particularly we saw with the orange data points, the vaccine effectiveness by time after the first and second dose still being high against symptomatic infection. Not quite as high as with alpha, but looking still high. And the hospitalisation data, the VE was right up in the around the 90% plus. Yes, over 90 for Delta. And even when Omicron came, we get around 90% effectiveness by this test negative method for VE with Omicron. And that was really important in us not locking down in Christmas 2021-22. And there was a lot of, because the Omicron coming, was concern about that. And then since then, we've been looking at the additional VE of booster doses on top of the protection you've already had. And what we're finding there, it's about, you get a sort of top up of around 50% further reduction with the boosters when they're given at least 3 months later, typically. But that does wane away with Omicron, as we've seen. And so that feeds into decisions about reboosting and so on. And VE is also demonstrated against other endpoints such as symptomatic infection and transmission from some of these other studies, and in different subgroups. So there was a lot of detailed data. On the safety side, this is a list of studies we've done in collaboration with others and speaking always to the MHRA and presenting to them. So the vaccine-induced thrombosis with thrombocytopenia, stroke, myocarditis, GBS, Bell's palsy, transverse myelitis, ADEM, anaphylaxis, mortality, and various pregnancy outcomes. The pregnancy outcomes are the ones we've been slowest on, and I think that is a lesson to learn around getting that not only did it takes longer because for outcomes you've got to wait for the vaccinations to be given and then the baby to be born and then to evaluate that. So it'll always be slower, and because this group got the vaccine later, that's why we're only now getting some of that pregnancy data through. It seems quite late, but that's how it is. It has been longer for that. So I won't go through all the results. From those that I've listed there, most of those I've listed because they were actually
recording in progress,
seen as events, but as true risks of some level. There were quite a lot of others we looked at with no evidence of associations. So finishing off on lessons learned and challenges, National Vaccine Registry with a population denominator has been essential to do these studies rapidly. It's been amazing really to be able to do things so quickly, and that proves so useful. Actually having multiple studies with different methods by different countries or even within a country is highly useful, because there's always could be critiques of any one method. It's when you're seeing different methods giving broadly the same results, it definitely adds confidence. So having that has been helpful, and that's where data from lots of different countries can be really helpful, and particularly things like the test negative case control has been very helpful as a rapid method. It's been essential to have good communications between all of the stakeholders. For VE, we had regular meetings of all those who were doing studies in the UK. So I spent quite a bit of time trying to contact everyone I knew who might be doing studies, pulling us all together on a Friday afternoon, regularly meeting, going through the studies and making sure we were on the same page. And I think that really helped for VE. For safety, I think it was a little bit less disjointed, a little bit more disjointed, perhaps from my point of view, in that I don't think from our side we didn't. I know, well, Catherine's going to talk in a minute, but information was coming into the working group on safety as well. But there were certainly some groups that I didn't know about that were doing safety studies, which caught me out and sort of surprised to find this suddenly producing a result on something I thought we were doing, for example. So in terms of challenges, keeping NIMS and other data sources going is going to be something that's really important, but it's not simple to get these things that have been built up going, because some of those who are administering these data sources might no longer see it as their job. That was just for the pandemic, and it's not up to us to keep it going and finding, but we want to keep it going so it's there, ready to go, not only for pandemic vaccines, but others like RSV, Group B strep would be great to keep. Understanding the importance of bias with the various methods with real-world data. I think that's still something that would be good to keep doing the research into, and to quantify and reduce that where we can. So what we can learn from that, there was a WHO call on Friday sort of talking about that quite a lot. And how real-world evidence generated by public institutes can be used by manufacturers for regulatory. Well, in a way, that's what these sorts of meetings are about. How can we join together to make sure we get the most out of the data between those of us who maybe have more access to it, those who can't access it so easily? How can we arrange either the studies to be done or data to be shared where that's possible? I think that was my last slide. Finish there. Thanks.
Okay. Any questions from the virtual audience?
Thanks, Nick. Fantastic presentation. I agree with all of your challenges, but the third one in particular put you on the spot. How do you think industry can access that kind of data, which is available in real time, to allow vaccines to be developed and deployed quicker, so to reduce the reliance on clinical trial data? Sorry to put you on the spot with that one.
I kind of see two ways around this. One is industry, the regulators and us, we're all at the same table, and if it's known that we can produce that information, particularly on effectiveness or using some of this laboratory data that we can access, and we can do it quickly, then it might be that their pharmacovigilance plan would involve us not knowing that we would do studies and sharing at a certain level information with the manufacturer as well for them to pass on. And in fact, that already happens with quite a few of the vaccines, where we've got agreements with manufacturers that we will feed into their data that they then use. So that's a sort of us doing it because we're going to do it anyway type thing. In terms of the manufacturers having direct access into the data, I think that's something we're I think to be looked at as we go forward, because a lot of the laboratory-based data we use doesn't get back, as we were discussing at lunchtime, and whether it's a bit of an infrastructure thing as much as anything, as well as getting in permission. So that is work to do, because I think at the moment there's certain things which would be difficult for the manufacturers still to do, using with the fine level of detail from the lab data in particular. So it's not an immediate solution there, but maybe there is a longer term one.
Okay. Thank you, Nick. Other questions from the audience or from the virtual audience?
Thank you, Nick. I'm just wondering if I correctly understand the NIMS is already built around for COVID. How do you see it, the application of the same resources for other vaccines?
We would like to see it sort of used.
Do you
much
think
more. Do we think it will be?
I know, because one of the challenges.
I don't know whether Julia's got better insight than I have. We would like it to be. Julia, any further comment?
No, because as you said, that there's one challenge is the resources, the funding resources is getting
It's
down.
a resource.
Now,
There's a resource implication. Who sees it as whose responsibility it is? Which at the moment is not entirely clear, and how much prioritization is given to it. So we want, from our end, we're pushing as hard as we can, but it's not just. There's a lot more other stakeholders.
Absolutely.
Yeah.
Any other questions from the audience? I'd like to ask one question which refers to something I have commented before. In an emergency, you make decisions. Sometimes you have to make decisions with insufficient data, incomplete data, and in some occasions you have to change the decision or go back in a decision. Could you give us an example of that in your case, in the case of your institution?
I mean, it's trying to come up with, for example, the age cutoff on the VITs, and I think that data just kept, that every few days we were getting more and more through on that. So I think that moved around quite a bit before. I mean, we didn't make. There was only one final decision, but in the interim few, even like a couple of weeks, there was a lot of discussion around that, and obviously in different countries. So yeah, I don't know about one where it's like the decision was made and then completely changed afterwards.
Maybe the interval between doses, as an example, and would you do it again? Would you wait and decide, or would you just make a decision and then
I think
modify?
For us at for JCVR, I think that was a fairly straightforward decision, really. Once all the information was put together, which was done quite rapidly around the modelling of doing that in terms of the number of hospitalizations and deaths averted, it was pretty clear cut. We just had to make sure that we then went back to the MHRA and said, this is what we're going to do. But there's always a bit of leeway in how once a vaccine's licensed, slight off recommendation things are done a lot. I mean, in the UK, it's not unusual for us to do that. So I don't remember there being a lot of debate, really. I think it was all pretty clear on that one for us. I know there was a big debate elsewhere. We teamed up with Quebec, I think, because Quebec were doing the same thing. So we had a partner there, which was nice.
Yeah, we had some debate ourselves, so we didn't have such an easy time. But thanks for your presentation and thanks for your contributions.
Thanks.
Okay, the next presentation is from Catherine Donegan from MHRA. She's head of AP in MHRA and PhD statistician. You have worked in HIV and also since 2010 in MHRA. And thanks for being here and presenting for us.
Thank you, Hector. So just before I start my presentation, after hearing Tom's excellent talk this morning and then listening to mine, you'll probably reach one of two assumptions. Either that we did not confer about the examples that we were going to present, or that we did confer and we've excellently come up with slightly different perspectives. But I'll leave that up for you to decide. So yeah, I think as Nick has already sort of helpfully described to you, the MHRA has a specific role as the National Competent Authority regulator in the UK, but we also have a statutory role in operating the vigilance system for all medicines, medical devices and vaccines. And so we really led on the UK's vigilance strategy for the COVID-19 vaccines, but obviously not in isolation. We had huge input from public health partners at UK HSA as it is now and the devolved administrations, and engaged a lot with academia and other experts. And we actually started planning our surveillance strategy and really thinking about the data needs that we would have, the methods we wanted to use, and how we were going to communicate on those data as far back as May of 2020, so a mere 2 months into the pandemic. And we brought together an expert working group, different to the one that Nick mentioned. We do like an expert working group at the MHRA. We'll have one for a lot of things. But this one was specifically around helping us design our surveillance strategy and helping us think about how we could make sure there was robust real-world data capture, how we could share that data and enable access to us as the regulator and other relevant stakeholders to conduct strong vigilance, how we could conduct continuous benefit risk assessment with advice from an alternative expert group that on the COVID vaccines benefit risk, how we should share assessment and regulatory decisions with the JCVI, also how we can publish our data to increase transparency and lead international engagement on safety. And there we particularly worked as part of the international collaboration of medical regulatory agencies, where we co-led that ICMRA COVID-19 vaccine safety group with the Australian TGA. So thinking a little bit first about some of the challenges for safety surveillance that real-world data had to help us address. Well, obviously, firstly, we were in a situation where with more limited follow-up in trials pre-deployment, and that obviously meant that we were going to have to ramp up safety surveillance. But one of the first challenges we had, as Nick highlighted, in the UK, that vaccines were targeted and prioritised for deployment. Firstly, to those at greatest risk, those with concomitant conditions and those much older populations in whom we had the least safety data. And we knew that it was going to be hardest to try and interpret case reports coming through in those populations and really understand what real-world data was telling us in that complex population. We'd have a very rapidly evolving evidence base due to the numbers of patients who are being vaccinated, and that we obviously the high public health need to deliver those vaccinations rapidly was because of the pandemic, and that pandemic was also having an impact on the healthcare system more broadly. So our ability and time of people, patients and healthcare providers to report to us, but it was also having an impact on presentation within healthcare services, diagnosis potentially, and incidence of some adverse events. Thirdly, I think over the months preceding the vaccine deployment, we were acutely aware of the public perceptions around the accelerated development and deployment of the vaccine. And we certainly in the UK saw a large anti-vaccine and more general anti-authority movement emerge. So more than ever, I think, whilst we were going to build a strategy that was really based around the existing infrastructure that we had, there was a need for more than ever, I think, for near real-time and proactive vigilance, which could really look at a range of modes and mechanisms of surveillance, different data sources, could help us with timely, consistent and credible risk communication, and could ensure that we were accessing data and sharing knowledge globally. So this slide just presents the kind of 4 strands of the MHRA's surveillance strategy. The 2 on the left-hand side are based around a yellow card scheme, which is the UK equivalent of the VAERS scheme, for medicines allows reporting of adverse events from patients and healthcare providers. And that we had 2 strands for that, both spontaneous reporting and also active surveillance methods. And both of those streams were really also supported by the collection of the vaccination data at the point of care, which Nick has already highlighted to you. We also worked with UKHSA and the NHS to ensure that we had good and robust and comprehensive point of care data collection on COVID-19 vaccinations, that was available not only in a single data set, but it was also fed back directly to patients. So you could get it in your NHS app, it was given to you on a piece of paper, and it was also sent back directly to your GP. So there was full transparency and awareness of the vaccine that you had received, which was really vital for supporting reporting across these other schemes. The other 2 aspects, the rapid cycle analysis we implemented in the clinical practice research data link. And again, prior to the vaccines being deployed, we established specific linkage of that primary care data set to the vaccination record data set and set up weekly feeds of data from the CPRD to ourselves. So we were able to look at a real rapid cycle analyses for a select number of adverse events of special interest. And then also finally, obviously the capacity for formal epidemiological studies, much of which was done on our behalf by UKHSA. So this is where we come to the overlap with Tom's talk. But the first, what I wanted to do was illustrate the use of some of these different sort of streams of real-world data for a couple of the adverse events of interest. And so the first one, thrombosis with thrombocytopenia, obviously a very rare event, initially identified as safety concern in February, March of 2021, but we had received a couple of case reports of very rare cerebral thrombosis earlier than that. And it's interesting to reflect on that, I think, a little bit and think about how our signal detection methodologies could have been more advanced to look beyond just the fact that those 2 cases had been reported, and maybe there was an opportunity for better looking at network type approaches to signal detection that may have picked up better a link with the thrombocytopenia earlier. As has been said, several countries halted the use of the AstraZeneca vaccine in March while the issue was investigated. The UK did not. This put us under a very sort of clear public and political focus, and there was a great need to act very quickly. I think early in the initial few days of this signal becoming apparent, it was very difficult to see that this was a new syndrome, and we spent a lot of time trying to understand what the rate of thrombocytopenia was with thrombosis. We established that thrombocytopenia was not well recorded in the data sets that we had, that interrogation of a hospital data set suggested that maybe as much as 40 to 50%, but that was highly variable, and really, I think, demonstrated that it was going to be incredibly challenging to use those kinds of data for this signal. So case reports were absolutely critical for this, and we spent a lot of time ourselves, UKHSA, working with clinicians across the country to try and ensure that we had a complete set, or as complete a set of cases as possible, so that we really start to talk about absolute risk. Obviously, that was then supported by bespoke studies, but it really did highlight to us that the more rapid admissions data is required. I'm running rapidly out of time, but I did just want to highlight one example of where we used real world data in communication. And I think this was a really good example because this was a slide that was actually presented on a press briefing to the UK around the time that we were describing the changes in use of the AstraZeneca vaccine and the switch to the Pfizer vaccine. And this was a slide that was presented alongside the University of Cambridge researchers there who really helped us put into perspective real-world data on the potential benefits of vaccination to different age groups, using vaccine effectiveness data and modelling data, alongside data from spontaneous reports to really help contextualise those benefits and risks. I'll skip over myocarditis in the interests of time and want to finish on some of the selected lessons that I think we've learned and some of the challenges that have been faced. So I think it's clear, and I think it's been brought up here, that case reports remain vital to effective vigilance. However, obviously they have their limitations, and we found that implementing rapid cycle analyses along those where we're looking at observed and expected within the same data set really could add an extra dimension to your sort of signal detection and signal refinement. However, the data structures are not necessarily in place for those kind of rapid analyses yet, and particularly around the identification of adverse events of special interest, where we've got delays in hospital admissions data becoming available, in the UK data not becoming available until the point of discharge, and even then that data is somewhat limited. Secondly, that the background rates and placing what we are seeing into context is incredibly important for signal detection, refinement, and also risk communication. But I think, use of these background rates in observed expected analyses needs much more careful consideration. It's not a one-size-fits-all situation, and I didn't talk about myocarditis, but there it really was a challenge to try and do any kind of observed expected, because our expected was very much based on hospitalizations, and our observed was a mismatch of all kinds of different potential myocarditis reports. Thirdly, and I think this mimics what's been said for effectiveness, that multiple studies with alternative designs and data sources are important, that good interactions with researchers and regulators are required. We certainly tried to do that, but the sheer number of research groups who sprung up working on vaccine safety was really quite staggering. But it was vital to have those conversations directly with researchers to understand data quality. And to try and reduce duplication. And then finally, I did just want to touch on perhaps one particular challenge that I think we found, and that was really understanding what data it was that patients wanted to help them make their decisions about the safety of the vaccine. And two sort of particular sort of signals or non-signals sort of really influence that. And I think one, it's been discussed a lot, it's around the safety of the vaccines in pregnancy. We hadn't anticipated, well, we should have anticipated better that we needed to build that data much more rapidly. But the second was really quite unexpected. It was around the reports of menstrual disorders that we received. We had huge spikes of reporting in menstrual disorders, heavy bleeding, delayed periods, etc., from women around their vaccinations. And I think this sparked an awful lot of concern about what the longer term impacts of this vaccination were going to be among younger women. And we needed to really remain much more alert to that. And that was a really interesting challenge for us, because it meant we had to really look quite different data sources and work quite closely with researchers who are looking at period tracking data, which is a complete new to us. So I just wanted to end on that note. Thank you very much.
Thank you. Thank you. We are short in time, so we will have time for just one quick question. Any question? I'd like to make one question. You observed that menstrual disorders were an issue. Would you have some lessons learned from that? Something you would do differently regarding that at this point in the next pandemic, if we are going to have the next pandemic, right?
Well, I think so I think the challenge is that we really had to reach out globally there to try and identify who might have data that could possibly help us look at this, because this wasn't in any of the data sets that we had considered. So we spent a lot of time talking to colleagues in the US, really, who were looking at some of this tracking data, trying to reach out to individuals who had potential cohorts of patients that might be relevant. But I think if to do this again, I think we really need to understand at the beginning better the quality of those data. I think there was a lot of uncertainty about, how much weight we could put on that evidence. We didn't know anything about it. And I think there's potential that the case reports could, dominate that conversation, which was not the right response because, we couldn't contextualize that in any way. So it wasn't the right data to use.
Thank you. Thanks very much. The next speaker will be Miriam Sturkenboom. Miriam has a PhD in pharmacoepidemiology. She's also a pharmacist and MPH from Harvard, head of the Department of Data Science and Biostatistics at Utrecht. Work also as a professor in Erasmus University, has, more than 400 publications as of today. And please get on, otherwise you will not be able to speak.
Thank you very much, Hector. So I was asked to talk about Vak4U and the lessons learned and the challenges. So for those, these are my disclosures. For those of you who are not living in Europe, maybe you didn't know the ADVANCE project, but it was a project that was actually initiated by or paid by the Innovative Medicines Initiative, and it was following the lessons learned on the H1N1 vaccine, actually the pandemic, where it was really needed that people would be working together. And this consortium actually established the vaccine monitoring collaboration for Europe in October 2019, just prior to the pandemic happening. So that we were ready at that time to work together. So it's an entity, it's working with partner organizations who can become a member. Those are research organizations or public health institutes. Currently 30 members, and the vision is to generate best real-world evidence in a collaborative manner. And we implemented in Vak4U the blueprint that was written based on the ADVANCE project by ECDC. So there's a lot of partners that have access to large real-world data. You see here an overview. Many data sources can actually be accessed. Not all of them had very good data on COVID. We'll come back to that. But in total, 100 electronic health records of 150 million persons can be accessed for studies. So what is the approach? Here you see the data. It is actually, we use a common protocol. It's a distributed network, so data stay local. The analytics go to the data, and it's a common analytics approach. So it's one script that's being sent, and then the results are being pulled. So here's the different data sources from all of these countries. Actually, they convert it into a common data model. We use the conception common data model. Then the quality is checked. There is tooling around that, which is called Insight. Then here's the semantic harmonization, which is done with a couple of tools with code list and phenotype libraries. And R is being used for the scripting and the transformation of the data into the final evidence, which then is uploaded to remote research environment and actually pulled together. We haven't been speaking a lot about the quality of the data, but within the network, we do quite a lot of quality checking of to assess whether the data is fit for purpose on many levels, on the population level, also on the outcome level and the covariates. But a very important one is also the coverage of the vaccine. We have seen in the US, it was really difficult to get access to the data that on the vaccinations. So what we did here is in each of the countries and the sites that are participating to look at the coverage and benchmark it against ECDC. So here are curves of uptake of vaccination in children, increasing age groups, and then compared to the ECDC tracker data. And you see that they all align actually quite well. So this gave us confidence that we could utilize the data. So Vak4U has been involved with many studies actually around COVID-19, still ongoing studies, especially for regulatory required studies, but also 4 that have finished, which were done for the European Medicines Agency. First starting with background rates for all the ASI, which were available from the SPEAK project that was funded by CEPI. Then the early COVID vaccine monitor, where there was near real time, or others call it rapid cycle analysis, comparing the rates after vaccination with the background rates. And then a 2-year project to be able to rapidly assess any signals that were coming up, and an effectiveness studies looking at heterologous versus homologous schemes. How is it conducted? How are studies done? I think what we have established after, with the testing of advanced, that what works really well is to actually rotate roles. So to have the whole network being able to participate in studies, not only to provide data, but also the expertise as being a PI or programmer. So it's not one center that is only conducting the studies. Actually, there's a working together and scaling up because people are ready and can actually take multiple roles. So when there is a request coming, there is the secretariat that assesses the interest of partners to participate and to take certain roles. And they together create the study team, which can vary from one study to the other, and conduct the study. And then we have an independent scientific advisory board with many of the key methodologists that look at all the company-funded studies to make sure that we do it well. So what we see here, what we have learned is that the rotating roles allow for rapid scaling up. There's also a challenge with it especially because we needed to scale up so rapidly, there was very little time to align actually between each other. So people needed to act, and now is the time for consolidation and making sure we harmonize the way that the work is being done. So I would like to share some results from the various studies that are finished. Background rates, they were made available quite rapidly to EMA. We'll also put online the Excel sheets actually with the rates and could be utilized by many of the stakeholders for the observed expected analysis, was working through this model. So what was really helpful was that there was this list of adverse events of special interest available actually from the SPEAK project. The challenge, what we noted, is that sometimes in some countries, especially during the pandemic, the access to data and the approvals to that lasted very long, especially in France, and for the Danish sites where it's also very difficult because there were so many people trying to use data to generate evidence that it causes a clot, I think, in the system. This is results on the near real-time monitoring of ASI, which was, or the rapid cycle, we didn't call it like that, but what happened is post-vaccination rates were compared to the pre-vaccination rates, and a couple of significantly increased rates were found, which have also been discussed and recognized signals. So what we noted there really is that we created a dashboard, but having crude rates on the dashboard is really difficult because of this very high channeled rollout of the vaccine. So a lot of false positive signals popping up initially. So you really needed to adjust very well for the confounding and the way it was prescribed. Cohort event monitoring was done across 13 different countries using different systems. Also there we used the same protocol and we created a common data model across these systems to then pull the data together, a total of 650,000 vaccinees included with primary data collections. And also that report is online for those of you who want to see it. So what was really learned there is that this infrastructure existed in 2 countries, was ready prior to the vaccines being rolled out. And all the other countries that were actually setting it up later were already too late. Like it went so fast that the approval system, once approvals were in place, actually most of the vaccinees or people were already vaccinated and could not be enrolled anymore. It is also a very resource-intense system. Some evaluation studies here, the myocarditis. We repeated analysis for the European Medicines Agency. So the first time was in November. They asked actually end of September. So at the Prague meeting in early November, the first results were there, and then they were updated repeatedly. And as we can see, the data here after those 2, of course, we have seen that is what you expect, but also AstraZeneca vaccine. So I think it is significantly associated. So the challenges, I think, here is that really we see that age and gender are important effect modifiers for some of these safety outcomes, and that still, even if you use a lot of data, the power to do that actually, and then also to look at brand and those is limited. And we see a lot of variability in these cutoffs. So to conclude, what worked? Advance was set up to create an ecosystem to be ready, to have data readiness, people readiness, and method readiness. And I think it was very helpful that PAC4U existed to do that and to support during the pandemic. There are a lot of tools that are being shared across the community. And this allowed for successful and timely response to questions from EMA and PRAC. What could be done better? I think the 6 monthly reports that were required by EMA were very challenging to do and actually did not allow a lot of time to have profound discussion about methodologies. And I'm happy we can have that now. Thank you.
Thank you. Thanks for doing a timely presentation. We have time for one question from the audience. Anybody?
Miriam Bobchum, congratulations. There are other parts of the world that obviously need similar systems put in place. What would be your recommendation? Would you follow the same model with Advance, et cetera?
Well, I think in Advance, what was really important is that we had this multi-stakeholder collaboration. So it had both academic partners, other private research organizations, but also vaccine manufacturers, ECDC, EMA, public health institutes. So all stakeholders were together to create and build this. And I think that was really needed to build some level of trust and to know each other and to create that readiness. So I think that was crucial. So I think an initial project to establish that would be required.
Thank you. Thanks very much. The next presenter would be Kat Polaers from P95. Kat is a PhD in statistics. She's a methodologist, head of data science for P95. She has worked in Advance, has initiated COVID Drive as well. And she's also a master in theoretical psychology, if others didn't know. So when she talks to you and looks at you, she's really taking notes and analyzing you. So be careful. Okay. Please proceed.
Thanks, Hector, for this very nice introduction. Yes, I will talk about COVID Drive. So it's a disclosure, but also actually an acknowledgement of all the partners who collaborated on this project. So this is the partnership. We are an open public-private partnership, also comes with a lot of challenges. I'm happy to talk about it. It's privately funded only, also comes with challenges. And currently we have 9 partners. It's launched in 2021 as a response to the COVID-19 pandemic and because Hector forced us to. And it's running until at least 2025. It was initiated to conduct brand-specific COVID-19 vaccine effectiveness studies and was actually a sequel, if you wish, of the DRIVE project that was running at the time to do brand-specific influenza vaccine effectiveness. But of course, there was no longer influenza at the time. So we thought like, hey, maybe we can do COVID-19 instead, but that was not allowed by European Commission at the time. So we set up COVID Drive in parallel to DRIVE. So we have set up several COVID-19 studies as regulatory commitments endorsed by the AMA, and we are looking to expand to other pathogens. COVID Drive is basically a network of hospitals. We have 44 hospitals. I think 13 or 14 of them are actively recruiting. There are also GP networks and population registers within the network. So what are our guiding principles? We are an open partnership, so partners can join the partnership at any time or also leave at any time. We do have a long-term perspective because we don't want all our efforts to go lost. We work between public and private stakeholders. We also share knowledge and collaborate with each other. And one of the main reasons why we are doing so is because we would like to optimize healthcare resources in data collection. Because very often different vaccine companies are fighting for the same hospitals to collect data for them. And of course, that's also not very fair. So this is the idea that, I mean, the data collection is open for all partners to join. This allows us also to share study costs between the partners and therefore set up larger studies. We are also still working on allowing data access for secondary use. So the main accomplishments, of course, I mean, it's a consortium agreement and the governance charter. It was quite a challenge to have a consortium agreement with 8 vaccine company partners at the time and all their lawyers involved. So that was a huge challenge, and I think we would never have made it without COVID-19 pressure at the time, but still it took us several months to get there. We also have a wide network of engaged study sites who are really eager to provide data, but we're also having challenges there in continuation of the funding. And we also have set up an advanced data collection and monitoring system. We have an ECRF, we have a research server, we do risk-based near real-time center monitoring, we have bi-monthly progress reports, we have a suite of quality control and analytical scripts. I mean, we really went all the way. We have a study documentation system with versioning control and also an experienced multidisciplinary team built over these last 2 to 3 years. So actually, we are ready to deliver high-quality epidemiological studies with shared data collection. So now I'm going to say a bit about our first study we implemented. It's a test negative case control design. It's, I think, very well-known design here for this audience. And we were going to look at product-specific COVID-19 vaccine effectiveness against COVID-19 hospitalizations. And we also invested quite substantially in obtaining information on the genetic variants. So we did additional testing. The protocol is also available at the NSEP UPass register. So this is how it works. It's a common data collection. So, but we have company or study requester specific adaptations. So it's common in the sense that we all collect the same data elements, but of course not all vaccine companies were having their product on the market at the same time. So I mean, they could start later or end earlier depending on when their vaccine was used on the market. So we have a common data collection, but as you see here, AstraZeneca and Janssen, they kicked off the study and they will end soonish with their study. Then Novavax took over and Valneva, and hopefully we will have another vaccine company partner who will continue and using this common data collection. So we have built a master protocol, and then of course company-specific adaptations can be made, like the definitions in the exposure, for instance, or the countries or time period, etc. So it's kind of modifications are of course possible. Implementation, we started recruitment in September 15, 2021. As I said, we wanted to start way earlier, but the discussions on the governance charter and the consortium agreement, they took a bit of time. It's of course extremely difficult and challenging, but yeah, so that was one of the main problems we overcame. Then countries, we initially started recruitment in Spain and then Italy, and then we scaled up. We are also now recruiting in Austria, in Belgium, Germany. We are setting up additional hospitals in Germany and additional hospitals in Italy. And today we have over 10,000 SARI patients recruited in the study. So we really have a lot of data. So what were we able to do with this data? They were used for regulatory submissions by 4 companies so far. We have the protocols, obviously, but also progress reports, interim analysis, and final reports. And we have a first publication by AstraZeneca on their first interim analysis. So going to the lessons learned, yes, we had done several proof of concepts before, but now we were doing this for real. So yes, different vaccine manufacturers can still work together and set up a common data collection for regulatory use. We had public partners and study contributors who were all collaborating. We were able to share experience and scientific expertise and also resources, so we could really agree on sharing the costs of the data collection among different vaccine manufacturers, which was also not a very easy exercise, of course. We were able to adapt our study following the changing COVID-19 epidemiology. I think we really have set up a very robust and high quality data collection system, and still we continue improving the way we are working. But for me, one of the main lessons learned is that we're quite out of the box. We don't easily match any of the company templates, SOPs, or procedures. It is a regulatory study, but we have our own way of working with the master protocol and company-specific adaptation, so we cannot just implement whatever they want, right? Because it needs to fit the master protocol, which was agreed by all companies. So it was a little bit of discussions within the different companies to make sure that the way we work was also okay for their internal procedures. Then we had a lot of discussions about the most appropriate quality assurance framework, and while we are obviously an observational study, we were often compared to a clinical trial and also a bit forced to follow GCP guidelines, which are very, very difficult for us. I mean, it took us a lot of time. It really made the work way more intensive, and of course also it's slowing us down. So that's one of my main challenges, I feel, is what's an appropriate quality assurance framework that's acceptable for all of us, including the companies and their internal quality people or external audits, because that's also an experience from the past. The DRIVE experience, we did an audit for DRIVE and we were compared against GCP, while we clearly said we are not a clinical trial, but still that's how we were. You know, there was the benchmark that was used, and that wasn't very fair, I felt. But anyway, so I think there's still work to do in this respect. Roles and responsibility was also very difficult because P95 was acting as a study sponsor on behalf of all of the entire consortium, obviously, but that's also not so easy for many of the companies to give away the study sponsorship for a study that's a regulatory commitment to them. So also here, it comes with trust, it comes with a lot of critical questions, and it's not, again, here we are very strongly out of the box. Then the shared study budget, of course, it's nice because it allows us to do bigger studies, but of course it depends on other companies leaving and joining. So the budget forecasts were not very stable, and it really needed a lot of flexibility from all the companies involved. This obviously led to long discussions which were slowing us down. I hope we have learned a lot from discussions so we can reuse this for the future. One of the main struggles we are currently facing is because it's private funding, right? So this is resulting in discontinuities in the resources we are having for data collection, while we are actually wanting to have a continuous data collection, as we have set up local study teams in the different hospitals and trained them. And we did all the effort, but now, of course, we are a bit running out of resources. So I hope we can sustain this because of the future vaccines coming and maybe the next pandemic coming. So that's it from my end.
Okay, any question for the speaker at this point? I will start with one quick question. In your opinion, which could be the next step for COVID DRIVE if you were to move to another vaccine?
RSV.
Thank you. Any other question from the audience? Thank you, Kat. Thank you. Now, we have a few more minutes to spare. So does anybody has any question for any of the presenters at this point? Anybody? Bob?
Yeah, Bob Chen, Brighton Collaboration again. So the TTS episode is always interesting and very challenging one in multiple levels. In trying to figure out how we could best learn for it for the future, when it's a AESI-X, where the astute clinicians did pick it up, and I think the hematologist community is to be commended for all the excellent work that they've done. In some ways, we were lucky to have Dr. Andres Quennecker from Germany to have been kind of an expert in this kind of very subdomain of hematology research, and being able to then kind of build on that knowledge. We probably, we may or may not have such luck for the next AESIX. And trying to think about how we can best take advantage of these different clinical and research subspecialty communities around the world. Perhaps we could just trust that humans being humans will kind of via their own social network cluster and somehow get that signal to the authorities. I'm just kind of curious if others have any thoughts about that domain.
Any of the speakers, particularly Nick, Catherine, others, would volunteer to answer the 10-minute question from Bob?
I mean, it was a challenge. We did work with some of the clinicians, and we wanted to sort of get better data than we were getting from our data linkage. So we kind of contacted, what was the network called? Hemstar network, to try and sort of get that information on looking at the thrombocytopenia levels and get that extra clinical verification of the cases. So that was a lot of work, though. I mean, that was not simple, not quick. It took us months to get that. And I think one of the extra things we need, actually some of the sort of extra laboratory type testing data that's available, that's sitting in hospital records, is not easy to get hold of. So that's another thing that could be better, to get that extra info on, the sort of detailed information to look at these sorts of events better.
I just wanted to nudge Catherine a little bit, because I had a question. You mentioned maybe briefly about looking in your databases, and you used the word network analysis. So I'm very curious if you could expand upon that, because I do believe that there are quite some limitations as to the way we do statistical screening of databases using MEDRA, a very clinically redundant language in which I think a lot of signals have the potential to be spread out over a lot of different terms, so it can be a bit easier. It's not a very clinically, it requires clinical interpretation, so I should say. So I'm very curious if you do believe there might have been an opportunity to see this signal in the spontaneous databases, even maybe before it came, I don't know, even if before it came to the notice of physicians speaking out.
Well, I'm not very clinical, so I can't help you on the interpretation. I mean, I think my comment there was, so, and I think I said in the talk, we did have 2 cases of CVST before Andreas Greenaker raised his signal. I don't know whether we would have been able to find those better. I think you're right. We are very, in signal detection, we are very singular event focused. Single event, single drug. And we haven't got, we are always looking to improve our signal detection methodologies, but we, within a regulatory agency, have to inevitably have a sort of one-size-fits-all type approach. And I think that, we don't know whether in this case something more refined might have picked something up. Equally, we don't know if something more refined may have picked up a whole load of other nonsense. So we just don't know, I think. But I think it would be interesting at some point to explore what methodologies could offer.
Okay, the time has is finished.
Yes,
Coffee
can I
break.
invite you for coffee or tea? We have half an hour, but I think it is important that we take some time because after COVID-19, we need to talk to each other and to do some networking. Please be back at in 25 minutes so we can start again sharp at 3:30. Thank you.